Hey guys, Miss Cote here, and we're just working our way through our gene expression and regulation unit. In the previous videos, we looked at the structure and function of DNA and how DNA can replicate itself during the S phase of interphase. In the next series of videos, we're going to explore the steps where the information in DNA can be used by the cell to build a protein. So let's get started. We've previously learned that DNA is made up of subunits of nucleotides, what we refer to as the A's, T's, C's, and G's. And it's the specific sequence of those A's, T's, C's, and G's that provide instructions on how to make the proteins that your body needs. This chapter reviews the process of making proteins, called gene expression, and it happens in two parts, transcription and translation. As scientists were working out the relationship between genes and proteins, you can read about Beadle and Tatum's one gene, one polypeptide hypothesis in your book. One issue came up. If DNA cannot leave the nucleus and proteins are made on ribosomes out in the cytoplasm, then how does the information get from one place to the other? The answer was RNA. RNA, specifically messenger RNA, is the go-between between the DNA and the proteins. Copying the instructions from the DNA to the mRNA is the purpose of transcription. Then, the mRNA can leave the nucleus, find a ribosome in the cytoplasm, and undergo translation, which is using the instructions to create the polypeptide. And of course, we need to compare and contrast prokaryotic gene expression with eukaryotic gene expression. And while both prokaryotes and eukaryotes perform transcription and translation, since eukaryotes are more complex, there's a little more to making proteins in a eukaryotic cell. For example, since eukaryotes have DNA that's contained in a nucleus, transcription and translation are two distinct processes. In prokaryotes, because the DNA is right there in the cytoplasm, the two processes can happen at the same time. Translation can start before the full piece of messenger RNA has been transcribed. And second, in eukaryotes, there's an extra step between transcription and translation. This is not something you learned in honors bio, but the messenger RNA transcript needs to be processed before it can leave the nucleus. We'll see what goes on there in a little bit. One phrase that you may hear a lot in molecular biology is the central dogma. This refers to the explanation, which is generally accepted in the scientific community, that the flow of genetic information in an organism goes in a certain order. We start with DNA, which produces RNA, which is then synthesized into a protein. Like anything we've been studying this year, there are exceptions to this rule, and we will look at one major exception in the virus video. So let's start with the process of transcription, the first stage in protein synthesis. In contrast to DNA replication, which we studied in the last chapter, transcription only involves one enzyme. RNA polymerase is able to separate the DNA and add in the RNA nucleotides all by itself. No primer is needed to start this process, and the nucleotides bind to their complementary base pair. However, please remember that there is no thymine in RNA, so instead of adding a T, you add a U for uracil in its place. The process of transcription can be separated into three stages. Initiation, which is everything that needs to happen for the process to get started. Elongation, the lengthening of the RNA transcript. And termination, finishing up when it gets to the end of a gene. So let's take a look at these three steps using diagrams. When we need to make a protein, we don't need to copy the entire chromosome, which could be hundreds of genes. We only need to make a copy of the instructions of the one gene of interest. Here we have a stretch of DNA. Notice that the two strands are designated non-template strand and template strand. The strand that has the instructions and will be copied is the template strand. Note that it can be either strand depending on the orientation of the promoter and gene. At the beginning of every gene is a region called the promoter, and somewhere in the promoter is a special sequence of bases called the Tata box. This is a series of T's and A's that helps RNA polymerase know where to come in and bind to the DNA. But before RNA polymerase comes in, other proteins called transcription factors bind first. Transcription factors are proteins that can either activate or inhibit protein synthesis. In this case, it's to activate transcription. The transcription factors in RNA polymerase together, attached at the promoter, is called the transcription initiation complex. At this point, initiation is complete, and we can move on to the next phase, which is elongation. In this second stage, RNA polymerase will move down the DNA to create the RNA transcript. It separates the DNA strands 10 to 20 bases at a time, just enough to add in the new nucleotides. This happens pretty fast, adding about 40 nucleotides every second. If you look at the diagram, the RNA nucleotides, shown in red, are added one at a time to the 3' end. Again, just like in DNA replication, new nucleotides can only be added to the 3' end. The directionality is important, and this elongation process will continue on until RNA polymerase gets to the end of the gene. The third stage of transcription is termination. Obviously, this is what happens at the end of the gene. It's a little different between prokaryotes and eukaryotes. In bacteria, there's a sequence called the terminator, and this is where RNA polymerase stops transcribing. And remember that in prokaryotes, ribosomes can start translation before the transcript is even finished. However, in eukaryotes, RNA polymerase will recognize a specific sequence, it's basically a bunch of A's, and stop transcribing a little after that. So at this point, we have an RNA transcript. I just want to emphasize that all RNA molecules are made in this way, whether it's messenger RNA, transfer RNA, ribosomal RNA, or any other type of RNA that we haven't learned about yet. But the only type of RNA that is translated into a polypeptide is the messenger RNA. But before it can leave the nucleus and find a ribosome for translation, we need to modify it a little. There are enzymes in the nucleus that modify the messenger RNA transcript. Don't worry, you don't need to know the name of those enzymes. This is called messenger RNA processing, and 3 things need to happen before the mRNA is allowed to leave the nucleus. Both ends of the transcript need to be altered, and we also need to get rid of unneeded nucleotides inside the transcript itself. So let's take a look at what happens. First, let's look at what happens to the ends. Here in the middle, shown in red, is our transcript. On the 5' end, we attach a GTP. We call this the 5' cap. At the 3' end, we add a bunch of adenines, and we call this the poly A tail. Scientists believe the reasons for doing this is threefold. It helps move the mRNA out of the nucleus and into the cytoplasm. Once it gets to the cytoplasm, the ends help the messenger RNA from being degraded by cytoplasmic enzymes. And last, the GTP is important for helping the mRNA attach to the ribosome. So that takes care of 2 of the 3 modifications. The third modification is to remove unnecessary nucleotides from the transcript. Most genes have stretches of nucleotide sequences that do not code for the polypeptide. These are called introns and are represented as the pink portions of the transcript. The red portions represent the part of the transcript that will be expressed to make the polypeptide. We call these exons. So to get rid of the waste and to make things easier at the ribosomes, the third modification is to edit or splice out the introns and join the exons together. As you can see in this diagram, this makes the mature mRNA transcript much shorter than the original transcript, and the coding sequence is now continuous. This mRNA is now ready to leave the nucleus. Now these introns just don't splice themselves out by magic. Of course, you need to understand how the splicing process works. Splicing happens through a complex of proteins, shown in purple, and small pieces of RNA, shown in red. Red pieces can sometimes be referred to as SNRPs. Together, this complex is called a spliceosome. The spliceosome recognizes the splice sites where the cutting and editing needs to happen. As shown in the diagram, the introns are cut out and the exons are connected together. The splicing is catalyzed by the RNAs. They are considered enzymes and are called ribozymes. So way back in biochemistry when I told you that all enzymes are proteins, well, I kind of lied. Sorry. As we're finding out in biology, there are always exceptions to the rules, and this is one of them. So here's another type of RNA you need to know about, other than M, T, and R RNA. So we've covered transcription and messenger RNA processing in this episode. I'll be working hard to finish the next video, which is the translation of the messenger RNA into a polypeptide. So until then, be a good human.